You know, when a mosquito lands on your arm, there's this natural assumption that the biological transaction happening is incredibly simple.
Right, yeah, like
Yeah,
a tiny flying syringe.
exactly. You think, okay, it takes a little blood, leaves a little itch, and well, if it happens to be carrying a pathogen, it just shoots it straight into your bloodstream.
It's a very mechanical way to look at it.
Yeah. But when you zoom in, like when you actually map the microscopic terrain inside that mosquito, that whole illusion of a simple delivery vehicle just completely shatters.
Oh, absolutely.
Because what you are actually looking at is this sprawling, chaotic biological war zone. And welcome to the deep dive, by the way. Today we are digging into a really mind-blowing paper.
I am so excited for this one. It's a massive study published in Nature in February of 2026.
Yeah, titled Mosquito Capsid Interactions Contribute to Flavivirus Vector Specificity, which I know sounds like a mouthful.
It does, but the core mission of this deep dive is to basically uncover the secret transit system that viruses use to travel inside mosquitoes.
Right. And to finally answer this huge, long-standing biological mystery, why do specific mosquitoes only carry specific viruses?
Exactly. And, you know, the stakes here are just difficult to overstate. We're talking about flaviruses, so dengue, Zika, Japanese encephalitis.
Basically the heavy hitters.
Yeah, the worst of the worst. Hundreds of millions of human infections every single year, and the entire global spread of these diseases relies entirely on a microscopic journey taking place inside a millimeter-long insect.
So let's trace that journey because I think people need to realize it is definitively not a straight shot from point A to point B.
Not at all.
A mosquito bites someone infected with dengue, right? The virus enters the mosquito's midgut along with the blood meal. But to infect the next person you bite, it can't just hang out in the stomach.
Right, it has to escape. It has to cross the gut barrier, enter the mosquito's internal body cavity, navigate the insect's circulatory system, and somehow successfully invade the salivary glands.
Because it has to get into the saliva.
Exactly. But, and this is what the study so elegantly reveals, that transit through the circulatory system should basically be a suicide mission for the virus.
Okay, let's unpack this, because the virus needs to make it to the salivary glands. But the hemolymph, which is the mosquito's equivalent of blood, is basically an absolute acid bath.
It is incredibly hostile.
So there has to be some physical mechanism protecting it from just melting the second it leaves the midgut. And I want to look at the environment of that hemolymph first, because the way the researchers measured it was just really cool.
Yeah, so they used these tiny microelectrodes and injected this fluorescent pH-sensitive dye, it's called HPTS, directly into live mosquitoes.
Wow, into living mosquitoes.
Yeah, and HPTS is fascinating because its fluorescent emission shifts dramatically based on the concentration of protons in its environment. So it lets them track the pH in real time inside the living insect.
That's wild. And what did they find?
Well, across multiple species, Aedes aegypti, Culex quinquefasciatus, they found the hemolymph is consistently, constantly highly acidic. The pH sits right around 5.9 to 6.1.
Wait, really?
Yeah, and it stays there even after the mosquito digests a huge blood meal.
To put that in perspective for anyone listening, human blood is super tightly regulated at a pH of 4.
Right.
So a pH of 6.0 in a biological fluid is incredibly acidic. And looking at the structure of a flavivirus, that specific acidity level is basically a self-destruct button for the virus's outer shell, right? The envelope.
Exactly, the envelope, which is made of what virologists call the E protein. And that E protein is the critical tool the virus uses to enter a host cell.
It's the skeleton key.
Yes, but it is inherently spring-loaded and highly sensitive to proton concentration. So when the pH drops below 6.5, the histidine residues on that E protein take on extra protons.
Okay, and that changes things.
It changes their electrical charge, which basically repels nearby molecular structures and triggers an irreversible conformational change. The protein forcefully folds outward.
Oh, wow. So it's essentially deploying its entry mechanism in an empty room.
That's a great way to put it. It springs the trap meant to fuse with the human cell, but it does it while just floating in the mosquito's blood, completely burning the one tool it has to ever infect a cell again.
The key is permanently bent. A free-floating, mature virus particle is just entirely disabled by the mosquito's baseline biology.
But wait, if the mosquito's own blood acts like a vat of acid that destroys the virus, how does the virus ever survive the trek to the salivary glands? I mean, it's like an armored car completely melting the second it hits the highway.
Right. It's a profound mechanical paradox. If the naked virus gets neutralized the moment it hits the hemolymph, it physically cannot travel naked.
It has to have a disguise.
It has to be shielded. And this is where the paper documents this brilliant evolutionary workaround. The virus completely ditches its own envelope.
It goes naked.
It sheds the very structure the acid would destroy. It strips down to its bare core, just its viral RNA and the capsid, which is the inner protein shell.
But a bare core can't just float through the acid either, right?
No, it still needs a transport vehicle. So it hijacks the mosquito's own extracellular vesicles, or...
I found the mechanics of these EVs so fascinating, because these are basically tiny biological cargo pods that the mosquito's own cells are constantly budding off, right?
Yeah, they use them to send proteins, lipids, and genetic instructions to other tissues.
So they are naturally designed by the mosquito to survive the acidic blood.
Exactly. They are the native internal transit infrastructure. By hiding its naked core inside an EV, the virus essentially stows away inside the mosquito's own cellular delivery fleet.
It's like a bank robber realizing their getaway car is recognized by the cops, so they hide inside a local Amazon delivery truck to bypass the security checkpoint.
That is exactly what it's doing. And what's amazing is how they proved this evasion was actually happening.
Yeah, I expected them to just look under a microscope, but they physically tested the vulnerabilities.
They injected neutralizing antibodies, which are specifically engineered to target and destroy the viral envelope straight into the mosquito's thorax.
And what happened?
The virus propagated perfectly. The antibodies had zero effect.
Because the envelope wasn't even there.
Right. The core was safely shielded inside the mosquito's EV lipid bilayer, but they went further to prove it using chemical inhibitors like GW48.
Oh, I looked into GWA69. It doesn't even target the virus at all, does it?
No, not at all. It targets a specific mosquito enzyme called neutral sphingomyelinase, which the cells need to generate ceramides.
And without those ceramides, the mosquito cell membranes physically can't curve inward to form and release those EV bubbles, right?
You've got it. So by injecting GW4869, the researchers effectively shut down the mosquito's internal delivery fleet.
And without the Amazon trucks?
The viral infection completely flatlined.
Yeah.
Without the EVs to hide in, the viral cores were exposed to the acid and destroyed.
That is incredible.
They even extracted EVs from infected blood, used immunogold staining to highlight viral proteins, and put them under an electron microscope. You can clearly see the dense viral capsids nestled safely inside the mosquito's vesicles.
Okay, so the virus ditches its uniform and stows away in the cargo fleet. But that introduces a massive logistical hurdle.
Oh, for sure.
Because a viral core can't just passively float into a delivery truck. The mosquito cell is carefully curating what goes into those pods.
Right, it's a controlled process.
So there has to be a physical loading mechanism, like a molecular tether, that specifically grabs the viral core and pulls it inside before the EV pinches off and enters the blood.
And finding that tether was essentially searching for a microscopic needle in a highly complex haystack.
How did they even start?
They isolated the EVs and ran them through a mass spectrometer to identify the contents. They found 437 different mosquito proteins that are naturally packaged inside.
407 suspects.
Yeah, and to narrow it down, they used this incredibly elegant technique called RNA interference, or RNAi.
I've heard of that. Instead of chemically blocking each protein, you sort of program the mosquito's own cells to stop manufacturing them.
Exactly. They synthesized double-stranded RNA that perfectly matched the genetic sequence of the top 30 most abundant proteins on their list.
And then they just inject that.
Yeah, and when you inject that into a mosquito, the cell's natural antiviral defenses mistake it for an invading pathogen. They chop it into small guide strands and then use those strands like a mugshot to hunt down and degrade the mosquito's own messenger RNA for that specific gene.
Wow. So they systematically turned off the genes for the top 30 proteins one by one, infected those mosquitoes with dengue, and watched to see if the virus still made it to the salivary gland.
It's a tedious process of elimination, but they actually struck gold really early on.
Oh, really? Which one was it?
When they silenced a gene labeled AAEL010585, dengue propagation was virtually abolished.
They found the loading mechanism.
They did. That specific gene encodes a molecule called VCP, or valucin-containing protein.
Okay, VCP.
VCP is a type of ATPase, an enzyme that normally uses cellular energy to sort native cargo into vesicles.
So it's the designated molecular tether, the bouncer at the club.
Exactly. Under normal conditions, VCP is just doing its job, grabbing mosquito proteins and dragging them into the EV so they can be shipped around the body.
But the dengue virus has evolved a capsid protein with a surface topography that basically mimics VCP's natural cargo.
Yes, the dengue capsid selectively binds to VCP, effectively hijacking the loading process. So when VCP gets pulled into the forming EV, it drags the bound viral core right in with it.
Okay, here's where it gets really interesting for me.
Yeah.
If VCP is the bouncer letting the virus into the club, does every mosquito have the same bouncer? I mean, is this why I don't get dengue from just any random mosquito bite in my backyard?
That exact realization is the absolute core of this paper's breakthrough. The structural shape of the VCP molecule is highly conserved across mosquitoes, but there are minute variations between species.
And those variations are the key.
Those tiny structural variations dictate vector specificity entirely. So let's break down the natural pairings. Aedes aegypti mosquitoes, they're the global vectors for dengue and Zika. Their native VCP perfectly conforms to the capsids of dengue and Zika. It binds tightly, loads the core into the EV, and the virus survives.
Okay, but what about a Culex mosquito? That's the one that carries Japanese encephalitis virus, or JEV. If a Culex mosquito bites someone with dengue, why doesn't the disease spread?
Because the Culex VCP has a slightly different surface architecture. It perfectly binds to the JEV capsid, but it completely fails to recognize dengue and Zika.
There's no physical bind at all?
None. The dengue core never gets dragged into the EV. It ends up naked in the acidic hemolymph, and it just degrades.
Wow.
And look at Anopheles mosquitoes, the ones that carry malaria. Their VCP structure rejects the capsids of dengue, Zika, ADJ.
So an Anopheles mosquito could gorge on blood completely loaded with dengue, and the virus will just perish inside the insect.
Exactly.
It's stunning how specific this is. You usually imagine vector incompetence is driven by some robust insect immune response, like actively hunting the virus down.
Yeah, an active war.
But in reality, it's just a missed physical connection. The lock and the key just don't quite fit.
It's pure structural chemistry. And to prove that the capsid was the sole deciding factor, the researchers engineered a chimeric virus, which honestly was arguably the most fascinating experiment in the whole paper.
The Frankenvirus.
Yes, the Frankenvirus. Using reverse genetics, the team took a dengue virus genome, cut out the genetic sequence for its native capsid, and stitched in the capsid sequence from the Japanese encephalitis virus.
So just to be clear, the virus is genetically dengue on the inside. It has dengue envelope proteins on the outside. But the inner structural shell, the capsid, is pure JEV.
Correct. And remember, a wild-type dengue virus cannot infect a Culex mosquito. The Culex VCP rejects it.
Right.
But when they introduced this chimeric virus into Culex mosquitoes, the virus propagated aggressively and reached the salivary glands.
Because the Culex VCP recognized the JEV capsid, it grabbed the inner shell, loaded the dengue payload into the protective EV, and completely bypassed the biological blockade.
They fundamentally altered the virus's host range just by changing its inner disguise.
That is wild.
But they went one layer deeper. They mapped the atomic structure of both the EDS and Culex VCP proteins to understand exactly why the handshake succeeds or fails.
I want to look closely at the chemistry there. Out of all the hundreds of amino acids in that protein, what is physically happening at the molecular level that causes the handshake to fail?
It comes down to just 2 amino acids.
Just two.
Just 2. The variation occurs at positions 723 and 728 on the VCP protein chain. In the Aedes mosquito, the one that spreads dengue, those positions are occupied by aspartic acid and asparagine. We call them D and N. But in the Culex mosquito, those exact same positions are occupied by glutamic acid and glutamic acid, E and E.
Aspartic acid versus glutamic acid, but they're both negatively charged amino acids, right? What difference does that actually make?
While they do share a negative charge, glutamic acid contains 1 extra methylene group in its side chain, so it is physically just slightly longer than aspartic acid. And that minuscule extension of atomic real estate creates what we call a steric clash. The slightly longer glutamic acid side chain physically bumps into the viral capsid, preventing the 2 protein surfaces from sitting flush against each other.
So the bond cannot form.
Exactly.
The lock is literally just a fraction of a nanometer too shallow for the key to turn.
Precisely. And the ultimate proof of this came when the researchers used genetic editing on the Culex mosquito.
Oh, they edited the mosquito itself?
Yeah, they targeted the native gene in the Culex mosquito and swapped out the sequence for the EE amino acids, replacing them with the DN sequence found in E.
They shaved down the molecular lock.
They did. By mutating just 2 tiny amino acid building blocks, they completely rewrote the biological destiny of the organism. that genetically edited Culex mosquito instantly became highly susceptible to dengue virus infection.
Because the steric clash was gone.
The clash was gone, the VCP bound to the capsid, and the virus was granted entry into the EV transport system.
You know, for everyone listening, it is crucial to understand why mapping a microscopic interaction like this matters outside the laboratory.
Oh, the implications are massive.
Because if we understand the exact atomic dimensions of this lock and key mechanism, we have a clear blueprint for intervention. We can rationally design small molecule drugs or compounds that wedge themselves into the AG's VCP binding site, physically blocking the dengue capsid from ever making contact.
And the genetic implications are even more profound for public health. We currently rely really heavily on population suppression, releasing sterile mosquitoes to crash local numbers.
We're just wiping them out.
But with this data, we could pivot to population replacement. Imagine engineering a strain of Aedes aegypti that naturally possesses the Culex EE mutation at positions.
You'd release them into the wild, they outcompete the native population, they still bite humans, but they would be entirely dead-end hosts.
Yeah.
You are effectively changing the locks on the local delivery truck. The virus enters the mosquito, fails to bind to the modified VCP, and just degrades in the acidic hemolymph.
Yeah.
You eliminate the disease without having to eradicate a keystone insect species from the ecosystem.
It is a phenomenal leap forward in rational, targeted vector control.
It really is.
But if we connect this to the bigger picture, the authors make a really vital point in their conclusion.
Was
Well, while they've successfully mapped how the virus survives the hemolymph and navigates the internal transit system, the upstream mechanisms remain largely opaque.
meaning the very first hurdle the virus faces.
Exactly. We know how it travels from the gut wall to the salivary glands, but how does it initially escape the hostile environment of the gut lumen and cross the midgut epithelium in the first place?
We don't know.
We simply do not know yet. The physiological environment of the mosquito's midgut during blood digestion is intensely complex. It's flooded with digestive proteases and physical barriers. How the virus evades destruction there before it ever reaches the hemolymph and hijacks the EVs is an entirely separate biological puzzle waiting to be decoded.
Which leaves you with an incredible thought. I mean, if the mosquito circulatory system relies on this crazy labyrinth of acid vats, hijacked lipid vesicles, and molecular tethers checking amino acid clearances, what kind of undiscovered bizarre biological traps are waiting for the virus in the mosquito's stomach?
It's a whole other world in there.
We tend to look at a mosquito bite as a simple exchange. But underneath that itch is an invisible, chaotic war fought over fractions of a nanometer, a microscopic conflict that literally dictates the trajectory of global health.
Couldn't have said it better myself.
Well, thank you for joining us on this deep dive. Keep questioning the invisible mechanisms around you because the smallest variations almost always hide the most profound discoveries. Catch you next time.